Hello students, welcome to chapter 13. We're going to be talking about genes, chromosomes, human genetics, basic structures, inheritance, more genetic stuff. We're going to be talking about how chromosomes change over time, how people figure this out, stuff like that. Okay. So first of all, when we talk about chromosomes in general, we can talk about how they're mapped on, how genes are mapped on a chromosome. So each pair of linked genes have a characteristic recombination frequency. So we already know about recombination in general. We talk about that in meiosis. Whenever genes are recombined in crossing over, so whenever they cross over, they recombine whenever they're switched. So recombination frequency between any two linked genes reflects the distance between them and their chromosome. So you can see the distance between two genes depending on how far they are apart on the chromosome. So you can see that based on what's called a map unit. So depending on how far one gene is from another, that is their map unit. So you can start from one end to the other, that's 0 to however many map units is, or centimorgans it is across. So that's based on Morgan, Thomas Morgan, the person whom we're going to talk about in just a minute, whom dealt with Drosophila, which is the fruit flies. Okay, genes near each other are called linked genes. Very near each other. They're so close that whenever recombination happens, they usually don't split. So these are called linkage genes. So these were discovered by Thomas Hunt Morgan, whom studied Drosophila melanogaster. This is a fruit fly, a very well-studied organism. This is a model organism that a lot of geneticists use, because this fruit fly has a very short life cycle. They undergo egg, larvae, pupae, adulthood very quickly. They can be maintained in a very small amount of space, and they can have a lot of genetic and phenotypic changes go on very quickly. Anyway, so many important discoveries in genetic, including sex-linked genes, were made in what's called a fly room. You can keep them in a specific room with mosquito netting, so they don't have to, they're not going to fly out. In test cross offspring, he found that a high number of parental phenotypes had a low number of recombinant phenotypes. So basically, he can test cross a lot of parental phenotypes with a low number of offspring with the same phenotype. So he can just basically come off with different phenotypes with experimental results having different results all the time. So he can have a good number every time. Okay. So first one, you have parental phenotype. You have a wild type with red eyes, and a double mutant with purple eyes and vestigial wings. So red eyes, normal wings, purple-eyes vestigial wings. And the F1 generation comes out with red eyes, normal wings, red eyes, normal wings, right? All flies were wild type in phenotypes and were heterozygous for both pairs of alleles. So when they come out, they have heterozygous, that means they're carriers. They have carriers for the purple eyes vestigial wings, right? They're heterozygous, right? So on the test cross, right, you have to have a test cross in order to see whether or not they have the carrier gene. You have to test cross that with the recessive. So when you do a test cross, you're going to come off with half the normal type and half the recessive, right? Well, fusion of one type of sperm produced the male, and 4 types of eggs produce the female, and generates the progeny of the test cross. So you have parental phenotypes and recombinant phenotypes, right? So you do have a half and half. So you have wild type, normal, purple vestigial, red type, vestigial, and purple normal. All right, so we know genetic recombination, but I'm going to go over it again. So genetic recombination is a process in which 2 homologous chromosomes exchange segments with each other by crossing over during meiosis, right? So you have that crossing over event where one switches with the other one. Depending on the distance in which it does it is your recombination frequency, right? Recombination frequency is a function of the distance between the linked genes, right? The nearer the 2 genes are, the greater chance they will become inherited together, right? So we have PR and VG, PR plus and VG plus. That's our 2 different genes and our 2 different alleles. So depending on how close they are together, which it doesn't look to be very close, is our recombination frequency. So recombination frequency there is not really that much. And there is a number for it. I think it has something to do with the CM value, centimorgan value, or the map unit value, but don't quote me on that. Next, we're going to talk about sex chromosomes. So we all know what the sex chromosomes are in our bodies, which is our 23rd pair of chromosomes, right? Females have 2 copies of the X chromosomes, forming the homologous pair XX, or at least the normal pair is. XX pair is homogametic, right? Homo meaning the same, gametic meaning the gametes. Males have 1 X and 1 Y chromosome, right? So me, I have 1 X and 1 Y chromosome. The Y is an incomplete X. By itself is a smaller version of the X chromosome. It does have different genes, it's just incomplete in some way. Giving males an XY combination, meaning heterogametic. Hetero meaning different. Genes located on the chromosomes other than the sex chromosome are called autosomic, meaning they have the same patterns of inheritance of both sexes. So every other chromosome is the same on both males and females, except for the 23rd pair. So when it comes to inheritance, you have the eggs coming from the mother are both X's, so you get X or X. The chromosomes coming from the male either have an X or the Y, so you get either X or Y, right? So if you put that into a square, you get X or X for the female, or X or Y for the male. So it's either XX or XY. It's a 50-50 chance. Talking about the human Y chromosome. Human sex determination depends on the SRY gene on the Y chromosome. So, early in embryonic development, structures that give rise to the reproductive organs are on the XX and XY chromosomes. So when the SRY gene becomes active, parts of the structures grow the testes, and other parts, including the, other parts. I'm not an anatomist. Hormones from the testes cause tissues for female organs to generate and tissues for male structures to develop. So, unless you have that SRY gene from the Y chromosome, you're not going to grow that. Okay. Other sex chromosomes. So, in insects, XX females will produce, and then you have XO female or males. They'll have no Y. In birds, butterflies, some reptiles, the males will have the homogenetic pair, and then the females will have the heterogenetic pair. For your attendance assignment, what I want you to do is research some other things, some other sex chromosome pairs. Okay? What other sex chromosome pairs can you find in animals? What other sex chromosome pairs maybe you can find in, other things, like maybe there's plants that have them, or smaller things. Okay? So we just talked about sex chromosomes. Let's talk about sex-linked genes. So genes located on the sex chromosomes are sex-linked. They're inherently different in males and females. So the alleles carried on the X chromosomes occur in 2 copies in the females and only 1 copy in the males. So this can cause some issues, right? If you have only 1 copy in the males, right? Let's say that you have a recessive copy on the X chromosome, but nothing on the Y chromosome to block out that recessive copy. Right? It's going to show. That recessive copy is going to show. Right? But if you have 2 X's and one of them is dominant, right? It's going to show over that recessive copy. Right? If one of them is dominant, it's going to show over that recessive copy. But if you have nothing here to show over that recessive copy, it's going to show. And if that recessive copy is disastrous or somewhat deleterious, it's going to be bad, right? So let's talk about this. So alleles carried on the Y chromosome are present in males, but not in females because of that dominant copy. Thomas Hunt Morgan proposed that this pattern was accounted for the inheritance of X-linked genes, or what's called X-linked inheritance. So he used the red wild-type eye color and a mutant white eye color caused by the recessive allele of sex-linked gene on the X chromosome. Right, and he saw the difference between those. And because the sex-linked gene on the X chromosome saw that recessive allele, he mostly saw that the males had the white eye color more than the females. So the normal red eye color was mostly on the females than the white eye color. And so he saw that it was mostly X-linked. So inheritance patterns in Drosophila. The transmission of white eye allele from the male parent of the male parent to a female child to a male grandchild is called crisscross inheritance. So let's see how this goes. So the transmission of a white eye allele from a male parent to a female. So if you have an affected father and it goes to an unaffected daughter, right? Because the X is going to be overshadowing that. And then the carrier mother, because it's now a mother, goes to the male grandchild. And you can see this down at the bottom. That's called crisscross inheritance. See that? Goes from father to daughter to son. That's crisscross inheritance. All right. Morgan also performed a reciprocal cross of a true breeding white-eyed female with a red-eyed male. So basically he just did a true breeding meaning that you had all, it's all white eye female. So basically both of the X's had the recessive trait with a red eye male, so it's unaffected father. Reciprocal cross, so you just did a white-eyed female with an unaffected father. Right? Just did a cross there. I'd recommend, if you can't follow what's going on down here, watch a video of it. There's a lot of videos of this crisscross inheritance. X-linked genes in humans. We do have family records of royal families, especially those that have diseases, bad genetic diseases. So family records, pedigrees, things like that. These are what X-linked diseases kind of tell us the history of these X-linked diseases. So X-linked recessive traits appear more frequently among males, right? Because you don't need, two of those X's for the recessive gene to come through, right? The females need 2 recessive traits, right? But the males only need 1. Males only need 1 copy of the allele on the X chromosome to develop the trait. A good example, Queen Victoria was heterozygous carrier for hemophilia. So if we see Victoria's offspring here, so Leopold, Duke of Albany, he had it. And then we go down: Leopold, Morris, Alfonso, Rupert, Alexis, Henry, Voldemort, Frederick. I mean, go on down the line. All of this. Alexandra was a carrier. Irene was a carrier. Victoria was a carrier. Alice was a carrier. Right? So you can see all of these going down the line to see how many had either was a carrier, or when they were a carrier had passed it to their offspring. So there's a different type of X-linked type of activation. This is called X chromosome inactivation. And this also occurs in females. The effects are equalized in male and female mammals, but mostly occurs in females. But this is dosage compensation mechanism, and it inactivates one of the two X chromosomes in most body cells of females. So, condensation process folds and packs chromatin of one of the X chromosomes into a tightly coiled state called a bar body. So one of the X chromosomes will actually just fold itself into a ball, right? Just randomly. It will stay inactive. And that same X will stay inactive for as long as that cell splits, goes through mitosis. If the two X chromosomes carry different alleles of a gene, that gene won't activate. The other allele will, but the one in the bar body won't. So as long as that cell splits, the gene that's active on the other X chromosome will continue to work, but the one on the bar body won't, for whatever reason. For some genes, the X chromosome inactivation produces recognizably different phenotypes on distinct regions of the body. So this is how you get different colors on fur, different colors on the skin. So white patches on your cat, like your calico cat. Chromosomal mutations that affect inheritance. So there's all sorts of these types of mutations that happen in nature. Chromosomal mutations are changes in chromosome number or structure or whatever, right? So changes in chromosome structure occur when the DNA breaks. The broken fragments may be lost or attached somewhere else, or attached to a different chromosome. Just depends on what exactly happens in the molecule. So deletion, some part of the chromosome just deletes and gets lost somewhere. That's deletion. It just completely deletes itself from the chromosome. And that type of genetic difference can mean a lot or a very little. It just kind of depends on what is deleted. It could be an exon, or it could be an intron. It could be a lot, or it can be a little of the chromosome itself. Genetics is a really weird process. And then there's duplication. Segment is broken from one chromosome and inserted into its homologue. So you can see here, you have DE, and then it duplicates that DE process. Most of the time, this process doesn't do a whole lot. Duplication process usually doesn't do a whole lot. It sometimes just creates more proteins. Sometimes it can do a lot, but it's not really that important. Anyway, translocation, a segment that is attached to a different non-homologous chromosome. So this is kind of different. You have one chromosome over here. It's basically you cut that out and then paste it to another one. We've kind of talked about this before, where you have just basically a cut and pasting onto a different area, but in mammals, this can change a chromosome quite differently. Adding genetics to the end of a chromosome, it's not very healthy for it. The other one is inversion. So whenever we see inversion, we see parts of the DNA actually flipping around. And it's actually flipping all of that DNA around. And this is actually very dangerous, because when it's reading that line of DNA, it's not actually reading it correctly. So it'll read it starting backwards, and it'll mess things up. So the next thing is changes in chromosome number. So whole single chromosomes can be lost or gained during what's called non-disjunction. Basically, the failure of homologous chromosomes to split during meiotic division. So during meiotic division, you're supposed to have the splitting of chromosomes separating apart, going to separate cells, right? Well, when they split, the spindle fibers are supposed to split them evenly. Sometimes that doesn't happen, though. Sometimes when they split, they don't split them evenly. They split into 3s or 1s or 4s. And some of them don't get some of them don't get, more than 1. Rather than 2, they get 1. Rather than 1, they get 3. Rather than, So during this process, they end up with extra or missing chromosomes. And the ones that come out are called aneuploids. Normal sets with normal sets of chromosomes are called euploids. EU is true. Aneuploids, ANEU means without true. Okay. So humans who receive an extra copy of chromosome number 21, they have Down syndrome. And sometimes this just happens normally because either something happens in the family and that just it's a genetic disorder, or sometimes it's just because it's just something with age or whatever. Okay. Another thing that can happen with non-disjunction is the sex cells don't separate differently. So sometimes what you can have is the egg cell separates where two of the chromosomes separate where you have two X's on one side and nothing on the other. So the sperm cell gets an X, because if it doesn't have an X, it's not going to live. It doesn't have DNA, it doesn't have information, right? It's not going to be able to survive. But if the egg cell doesn't have information, it can still survive. It still has stuff in it. It still has, it's big enough that it still has stuff in it. But if the sperm cell doesn't, it's too small to actually survive. Anyway, if the egg cell splits and it doesn't have DNA in it, it can still survive long enough so that the sperm cell can get inside. Now, that being said, if the sperm cell gets inside and it has a Y, it's still not viable, because that's still not enough chromosomes in order for it to survive past a certain point. But if the egg cell gets in and it has an X, it will survive to a certain point, and it turns into what's called Turner syndrome. I'm not really aware of the effects of Turner syndrome, but it'll go into Turner syndrome. Anyway, so after that, if you get into what's called double X chromosomes, and that's when you have two X's in one XL, or two X chromosomes in one XL, and it gets fertilized by Y. That's Klinefelter syndrome. And then if you have the two X's and it gets fertilized by an X sperm cell, that's triple X syndrome. Changes in sets of chromosomes is lethal by mammals, but not by plants. Plants specifically can go through this. Mammals cannot. This is lethal by most animal species, except some wasps, ants, and bees, and other things. So monoploids have one set of chromosomes. Basically, if you're monoploid, you're also haploid, right? Individuals with more than the normal set of chromosomes are called polyploidy. Okay, so normal set meaning if you have more than diploid sets, if you have more than two. But this often results in a failure of the spindle to function normally. So you're going to have more than two, you're going to often result in that spindle to separate those two out, right? You're going to have three, well, it's not going to really be able to separate those three. You're going to have four, you're going to have a lot of more difficulty for those to separate. Now plants, they found a way to do this, but they don't have the normal way of separating their spindle fibers. They don't have the normal centrosomes, other things like that. Okay. Polyploids are often hardier and more successful in growth. Corn, like, they have like 8 to hundreds of different ploides in their cultivars, and you can even look it up. I know that watermelon can have up to 16 ploidy. It's weird. Like, the bigger they are, the more ploidy they are. It's crazy. Basically, if you have a larger fruit, if you see a larger fruit, it's going to have more chromosome sets. So if you think about it that way, like, most of the fruit at the store is genetically different than anything that you're going to see in the wild. Okay, so triploid has 3 sets, like tri is 3, and then tetraploidy has 4 sets, tetra. Hexaploid has 6 sets. Triploid, pentaploid, heptaploid, thinking nonaploid. Anything that is odd numbered, you're probably not going to get a good offspring. And there's a reason for that. Think about the meiotic separation events. If you have an odd number of chromosome sets, how is that going to separate evenly? It's probably not. You're going to have some problems separating, right? So most of the time, odd number ploidy anything is going to be what's known as a sterile ploidy. So if you have a triploid organism, you're going to have a sterile organism. And most of the time, if you have a triploid or pentaploid plant, the farmers keep it that way so that they can keep it sterile. There's a few other human genetic conditions caused by what's called mutant alleles in the chromosome alterations. And these will follow Mendelian inheritance. So one is called autosomal recessive inheritance. We've kind of talked about this before, but basically you have both the father and the mother as carriers, and then they're symptom free, but when they have a child, some of their children are going to be affected, right? So sickle cell anemia, cystic fibrosis, PKU, these are all examples of recessive alleles through autosomal recessive inheritance. Autosomal dominant inheritance. So you're going to have one of the parents that are affected because they are dominant for it, right? They'll have either heterozygous for the dominant trait or whatever, and then the other one is homozygous, and then their children will be affected somewhat because they'll inherit the dominant trait. Achondroplasia or progeria are examples of this trait because they have the dominant allele. X-linked recessive traits. These appear more frequently among males, right? Because it's X-linked, right? Because they only need one X chromosome to develop the trait. The females must have both. Red-green color blindness is one of these. Hemophilia, Duchenne muscle dystrophy. So if the carrier mother is heterozygous for these, they can be affected. If the father is affected, most of the time you're not going to get anybody affected, but then the daughters can be carriers. X-linked dominant traits. There's not really a whole lot of these going around, but there's a few. So one example is constitutional thrombopathy. Not really sure what that is. But basically you have, if you have the father affected, you can have both, you can have the daughters affected, right? Because you have the X-link that is passed down to the daughters. But if the father's affected, it's not going to affect the sons because, you know, the father's giving the Y chromosome to the son. If the mother's affected, though, there's a 50-50 chance of the offspring getting it, because it can either pass through one of the X's or the other X, depending on whether it's heterozygous or homozygous. Okay. Another is hereditary faulty enamel or dental discoloration. Okay. So, what can you do about it? In the book, it says you can go through genetic counseling. I'm guessing that this is something that costs a lot, but anyway. Genetic counseling allows prospective parents to assess possibility that they might have an affected child. Sure. But you have to go through your own family history. Counseling begins with identification of parental genotypes through family pedigrees or direct testing for altered protein or DNA sequence. Yeah, you'd have to check your own DNA. So you'd have to do a DNA test. With this information, counselors can often predict the chances of having a child with certain traits in question. So cancer, progeria. If you have any genetic disorders yourself, you can see if your child is going to have those disorders. 1% to 3% of all newborns have mutant alleles that encode defective forms of proteins required for normal functions. Yeah, that's coming up a lot more often, but it's not something that's too noticeable until later on. So some other things that you can try doing: prenatal diagnoses. These are something that is coming up a lot in modern medicine. So in prenatal diagnoses, cells from a surrounding embryo or surrounding tissues are tested for the presence of mutant alleles or chromosomal mutations. So this is amniocentesis. So they just take some cells from the surrounding area, from the mother's uterus. They analyze those for DNA or any type of mutant alleles. And they can test for more than 100 different genetic disorders, including Down syndrome, any type of chromosomal disorders, stuff like that. In chorionic villus sampling, cells are obtained by embryonic portions of the placenta. So you can also test around the placenta. The procedure is usually done about 12 weeks of development because of the risk to the fetus. So you don't want to stab anything in there. That'd be gross. So another thing after the birth is genetic screening, which tests are routinely applied to children and adults or newly born infants in the hospital. It can test inherited disorders early enough to start preventative measures before symptoms arrive. So like PKU, this is something that they can test before the symptoms start. Especially if cancer runs in your family. Some of the last things, cytoplasmic inheritance. Inheritance patterns of DNA found in the chloroplasts or mitochondria. Basic things like ATP generation. These all come from the female side of the family. This is all done from the female because the egg is much larger, right, than the sperm. The egg carries all of that with it. This is called uniparental inheritance. All progeny have the phenotype of only one of the parents, right? Maternal inheritance, right? These do not segregate based on Mendelian segregation. Basically, it's all from the female. It's not from the male, it's all from the female. It's all ATP generation. It's going to be also any cytoplasmic genomes, anything in the cytoplasm that has to do with DNA. Last thing, genomic imprinting. Researchers have identified about 30 genes in mammals whose effects depend on whether an allele is inherited from the mother or the father. So this is called genetic imprinting. So this depends on whether it's from the mother or whether it's from the father. The silent allele, inherited allele that's not expressed, is called an imprinted allele. So over here we see a maternal chromosome and a paternal chromosome. So this mouse has what's called an insulin-like growth factor, IGF-2. So that's IGF2 gene has an effect on size of this mouse. And this mouse will mate with another mouse that has that IGF2 gene, right? So the maternal has an IGF2 gene that has a normal gene, right? The maternal copy is imprinted, okay? The heterozygote inherited a gene, IGF2 gene from the male parent, develops into a small mouse. Okay? Only the paternal inherited gene has effect on size. But the maternal copy is imprinted. So in a mouse heterozygous for the normal gene of IGF2, the paternal gene is active, and the maternal gene is imprinted, so it's silenced. So if the maternal gene is silenced, right, the silent gene is inherited, but it's not expressed. As long as the normal gene is inherited from the male parent, the mouse develops into a normal-sized adult. See that? So even though it's imprinted, even though it's there, right? Even though it's still coming in, the paternal gene is coming in as well. It depends on where it's coming from. Okay, let's say that again. The normal gene is imprinted, right? But it's silenced. So the active gene coming from the heterozygote, from the female parent, develops into a normal-sized mouse. So in a mouse heterozygous for the normal gene, the paternal allele is active. So even though it's imprinted, it's there, the paternal gene is still active because the imprinted gene is silenced. So it all depends on where it's getting the genes from. Whether it's getting from the maternal or the paternal chromosome. Okay. All right. I suggest that if you didn't get some parts of this, go watch videos. Go watch some videos. Go read the book. All right? Our exam is going to be coming up here soon. We're going to be going over the review on Wednesday. Okay? Keep in mind that our lab practical is also Wednesday. Alright? Lab practical number 2. I'm going to be grading those today. The first one today. I graded a few. Graded a few yesterday. Alright? Make sure we're going to be going over some of the lab practical stuff today, right? So keep that in mind. All right, I'll see you later.